Good evening and welcome, everybody. Tonight, it's my pleasure to introduce to you Dr. Art Chadwick. He has a PhD in botany, I believe, molecular biology, but has taught everything from botany to paleontology, geology. The list goes on, and he's published in most of these areas as well. But one thing that really sets Dr. Chadwick aside is that he not only loves to teach, but he also loves the pursuit of truth. Does anybody here like science tonight? Science is one of the most exciting adventures. And I think what you're going to find here tonight and in coming presentations is that we don't have to believe science versus the Bible. I think what you're going to find from Dr. Chadwick's compelling presentations, both tonight and tomorrow, is that science actually does support the biblical narrative. I can't wait to hear this presentation tonight. I'd like to welcome my friend and somebody who I have immense respect for, Dr. Art Chadwick.
Thank you. Well, good evening. I feel too tall up here, so I'm going to step down. Tonight, we're going to start out with the idea of what evolutionists believe about life's origin. And I promise you we'll get back to that. But before we go any farther, I want to establish some principles, some philosophical principles that underlie all the science that I'll be talking about. Some of the things I'll warn you tonight are fairly science-y and may not exactly be easy to understand for some of you. But if you'll just bear with me, we'll get through them and get to the point, which is that we do know whether science is in favor of the Bible or not. We're going to talk about some issues tonight about origins and about whether evolution can explain what we see in the world around us. Hopefully, all of you, by the time we're through, will be convinced that evolution cannot explain the world that we see around us, if you don't already know that. And then tomorrow, in the morning, I'm going to tell you some actual science stories that we have done in pursuing our understanding of the history of the world. So we'll talk about the Yellowstone fossil forests and some other things. And then at 11 o'clock, I'm going to tell you something about the scripture that happened in 1844 that I'll bet you don't know about. And that's saying a lot in the Adventist audience, but for many of you not Adventists, and those of you who are not Adventists will appreciate hearing this amazing story that comes out of the Bible that God intends us to understand and share with the world. And then in the afternoon, I don't know what time the meeting's set for. 2 o'clock. Yeah, after lunch, I'm going to tell you about our dinosaur project in Wyoming and what an exciting project it is. This summer, we opened our new facility for the first time, a $2 million building on this ranch in the middle of nowhere. You're driving down this rutted, bumpy road, and you turn a corner, and here's this beautiful, beautiful facility that will enable us to continue our work into the future, long after I'm gone. So we'll talk about that tomorrow in the afternoon. So tonight, I'd just like to start with a discussion about what evolutionists believe, because if we understand what evolutionists believe, then we can understand why this conflict exists between creation and evolution. And so first we're going to go through a little philosophical study. There are things in the world around you that favor what I call the standard model. The standard model is evolution over millions, hundreds of millions, billions of years. So that's what I mean when I say The standard model is what I mean when I say the evolutionary model. I use those two interchangeably. The correct way to say it is the standard model. This is the model that many scientists adhere to and believe in. There's a reason why they do that. Because many scientists are committed to a naturalistic explanation of origins. Naturalistic means no God. That's the simplest way to put it. Many scientists subscribe to a philosophical tenet that you cannot have God and do science. I beg to differ with them, but I think that they are very committed to this worldview. Those people would give this kind of evidence that you see here on the screen as reasons to support their perspective, but it's really hollow. It may be challenging to you or to them or to us, but it's really hollow because there's this slide. Issues that support the concept of a fiat creation. Fiat creation means God created in 6 days a few thousand years ago. So we're comparing those two models. Some people try to blend them together to make some other kinds of models, but they really don't fly for good reasons, good technical reasons. But the two models that we're discussing are God did it, as he said in Genesis chapter 1, or the Bible is irrelevant and has nothing to tell us about how we got here. Those are the two models. So this is a list of things that are a problem for the evolutionary model. Origin of life. What was that first slide? How evolutionists view origin of life. Origin of life is a serious, serious problem for evolutionists. They don't have any way of explaining how life could have gotten here that makes sense. The fossil record. Now, the fossil record is usually used by evolutionists to support their perspective. But there are things in the fossil record that definitely do not support the evolutionary viewpoint. For one thing, when we see life appear, it appears suddenly full-fledged, mature, just the way we know it today. There is no part life or part creature that gradually changes into something else in the fossil record. That is pretty much explicitly true, and evolutionists will acknowledge that. And secondly, when you find modern kinds of life forms in the fossil record, they're all there at the very beginning. There are representatives of every phylum. Phylum is a big grouping of animals that biologists use. There are representatives of every phylum in the very first layers of rock that we find that have these fossils in them. And from there on, it's just a change in diversity, or a lot of it has to do with how the animals got around. Obviously, if you're in the bottom of the ocean when you start making the fossil record, you're not going to find humans down there because generally we don't live in the bottom of the ocean. You're going to find them somewhere else up higher. Then there's the fossil record in rocks. When you go out and look at rocks and the hillsides here or anywhere else you choose, you will always see that the sedimentary rocks are in layers, discrete, distinct layers. Anytime you see layers of rock preserved in the fossil record, it tells you two things. Those layers were deposited rapidly and they were buried quickly because they didn't have time to sit around for burrowing animals to work through them, or roots of trees to grow in them, or other things like that that would have disturbed it. Instead, in most cases, you'll find that these layers are undisturbed. That's in favor of the creationist model. Just as we look at the fossil record, there seems to be a general absence of evidence of time. That's a discrete thing that we can see, that we can use as support for the fiat creation. And then we get to molecular biology, and I know I have a whole talk just on that. Molecular biology argues adamantly that life had to have an intelligent origin and it had to be instantaneous in time. So these arguments are very strong support for the idea of the Bible, of fiat creation. When we actually try to balance them out, what we see is something like this. You might dispute with me about the nuances here, but this is basically what we see. Not this, not that, but something like that. And all of us know enough of mathematics to know that if you have the same thing on both sides of an equation, what happens? They cancel out. So no one can come to me who knows the data. No one can come to me and say, well, I believe in the naturalistic model, the standard model, the evolutionary model. I believe in that model because of the data. No, they can't say that because they're only looking at half the data, if that's the case. They can't do that. Why do they believe what they believe? It's because of some philosophical commitment to a model. So they have said somewhere in their career, naturalism is the only thing that can work in science. So therefore, that's my model. I accept it. That's my religion. So if your perspective on origins comes from this old book called the Bible, you're in pretty much the same boat as someone who says, well, this comes from the old philosophical system. Does that come across okay? Do you understand what I'm saying? Everybody's in the same boat here. You cannot tell me the scientific data forced me to believe in that because you're only looking at part of the scientific data. The other scientific data, and we'll talk about some of that tonight, suggests quite the opposite. Okay, so let's go back and talk about what evolutionists teach about where we came from. Are you all good with that? If we're alive, right? You're alive, I'm alive. How did we get to be alive? Did we come from a rock? From a little warm pond somewhere? Is that where we came from? We're going to talk about that. Evolutionists tell us life came from a series of accidents. No purpose, no plan, no goal. It may have taken a while, maybe even 1 billion years or more of accidents before something alive that is capable of self-replication came out. It would ultimately have had to have been made of proteins. Now, proteins we're going to talk a little bit about here. You all know what proteins are. Proteins are those things you have to eat to maintain your strength. Proteins are made of little units called amino acids. Amino acids, there are 20 different ones, and how they're put together determines what the protein is. Proteins all differ, different kinds of proteins all differ in the order and number of amino acids that make them up. Now, if you looked into nutrition at all, you know amino acids are one of the things that some people tell you need to take. But amino acids are plentifully available to you in your diet. And all cells are made of protein. All cells are made of protein. It doesn't matter what kind of cells they are. So if you're eating plants, guess what? Their cells have protein in them. If you're eating animals, they have protein in them. Protein is present in all cells. So don't let anybody tell you, well, you have to eat animals in order to stay alive because you need the protein, because I don't eat animals and I'm alive. So apparently that's not a true premise. Anyway, we're going to talk about proteins. But proteins are where you have to get to from mud in a small warm pond. You've got to get to proteins before you can even begin to talk about getting to life. All right? So, how do evolutionists visualize this happened? Somewhere, there was a planet called planet Earth. On this planet, there was a gaseous atmosphere that contained methane gas. That's like, similar to the gas. Well, if you use natural gas in your stove, that's methane gas. Water, you all know what water is. Nitrogen in the form of ammonia. You know what ammonia is? Some of you use that for cleaning. It's a really good cleaning agent. It's very basic, so it takes off acidic materials. Hydrogen gas. Now, hydrogen gas, you all know what that is? That's what the sun's made of, is hydrogen gas. And that's why we get warmth from the sun, because it's burning. So hydrogen gas, ammonia, water, and methane. 4 ingredients that scientists told us that the Earth, the primitive Earth, had in its atmosphere. Why did they choose those 4? Why didn't they choose some other gases? Because if any gas had oxygen in it, it would destroy all the other ones. So they couldn't have anything with oxygen. So they took these 4 gases, they built an apparatus that you can see here. It consists of a closed circuit, and one side is being heated, so it makes a gas out of the liquid. The liquid is water with ammonia dissolved in it, and then they put in methane gas and hydrogen gas. Very explosive. Yeah, that would blow up. In fact, the space shuttle that blew up, you know what that was? That was hydrogen gas that blew it up. And so hydrogen gas is very explosive, so they have to keep, what do they have to keep out of this apparatus? Oxygen, that's right. Any oxygen in there, and you would have a wonderful explosion in your lab. So they pass these gases around that tube and into the chamber on the left-hand side. In that chamber, a spark is passed through those gases as they go by. That's very important. They pass that spark through, and then they take the spark away. Because if they allowed the spark to keep sparking, they would get nothing. They would just destroy what they made. But when they did this and they let that experiment run, and then they trapped it down here at the bottom, see if this works. Oops. I don't know if that works. Anyway, at the bottom there's a trap there. They let it run for 20 days, and then they checked to see what was in that trap at the bottom. And you know what? They found some amino acids that hadn't been there before. There were amino acids that they had made by sparking this mixture of gases in that tube. That was amazing. They declared they had solved the problem of creating life from non-living material because they made some amino acids. How could they do that? Well, because they assumed if they made amino acids, then they can make proteins. If they made proteins, they can make a living cell. Okay, we're going to examine that. Here is an amino acid. This is a very simple structure. It has an amino group on the left-hand side, an acid group on the right-hand side, and then there's a little R there. For most of the amino acids, the only thing different about it is the R group. And here's a picture with some of these amino acids. It's not really important that you know these structures, but do know that each amino acid is a little different from the other amino acids. And this is going to play into the story as we try to build a protein from these amino acids. Uh-oh. We already got a problem. In fact, we already have 2 problems. Number 1, in those experiments where they made the amino acids, they only got a few of the 20 that are present in all proteins in your body. They only got a few of those. What are they going to do to make the rest of them? Well, they had to drop back and punt. They had to say, well, okay, we get these amino acids from this reaction, and then we have to do something special over here to make this amino acid, and something special over here to make this amino acid. We add some extra ingredients to make this amino acid, but we can't have those ingredients in there for the other amino acids, so they're cheating. There's nothing like the Earth, the primitive Earth, in that experiment. There is no chemist around to make all these things, is there? Well, if there's a chemist there, you've already made life, right? We assume. There's another problem. Each of those amino acids, with the exception of one that is the smallest one, each of those amino acids has a carbon in it, and that carbon can have 4 different groups attached to it. They can be attached in 2 different symmetries, as you see there. The blue one is on the right in the left-hand picture, and on the left in the right-hand picture. You cannot rotate that any way to superimpose those 2 images. Do you see that? Those are different. They're called stereoisomers or enantiomers or enantiomorphs, and those 2 chemicals are made in equal amounts when they synthesize these amino acids. They're made in equal amounts. All living systems only use one of those, called the L amino acid. Levo, it means left. I won't explain why. But anyway, only one of those is ever used in protein, and it's always the L amino acid, but you're making equal amounts of D and L forms. So how are you going to purify those so you only have the L ones? Nobody has a clue. So even if you could make all the amino acids, there is no way to separate the D from the L. Their physical properties are identical, so they would not sort out in any physical process that we can think of to separate them. Now we join those together into a protein. One amino acid, oops. Does that work? Yeah, there. Okay, there's one amino acid, and a third amino acid up there. So we've joined 3 amino acids together. To do that, we make 2 peptide bonds. Those peptide bonds are not like any other chemical bond. Normal chemical bonds, you can rotate the structure around the bond. Those you can't rotate. That's why proteins are proteins. Because by not making that bond so you cannot rotate around that bond, you're going to determine the shape of the resultant protein molecule. So if you join together these amino acids in a sequence, that sequence predisposes that protein to have a certain shape. And the shape of that protein determines its functionality, its activity, what it can do in your body. What do proteins do in your body? Well, first of all, they're structural. They make up your body. Secondly, they are functional in that they carry out the reactions that you need in order to stay alive from moment to moment. All the things happening inside your cells right now are being mediated by proteins called enzymes. And these enzymes are made of amino acids joined together in a specific sequence that take a specific shape that will carry out a specific reaction to make sugar turn into energy in your body. Does that make sense? If you change one of those amino acids in that protein, it's going to have a different shape, and it's not going to do what you want it to do. Here you can see in the upper left what's called the primary sequence. This is the sequence, in the pictures in the upper left. That's the primary sequence is the sequence of amino acids that you've added together to make that protein. So you add one amino acid, and you build this big long chain called a protein. That order of amino acids determines which protein that is. That then predisposes, that order of amino acids predisposes that protein to form into the structures you see on the right-hand side. And those structures together with the parts that aren't formed make up the tertiary structure of the protein. That's the shape of that particular molecule of protein. If it's any other shape, it won't work. Do you know what? Any of you ever heard of prion disease? What's another name for that, Josh? Mad cow disease. Prion, or mad cow disease, is a normal body protein with all the amino acids in the right spot that's folded wrong, and you die. So the folding of these proteins and the order of the amino acids are exquisitely important to your survival. The one in the lower right is the other thing that proteins do. Besides carrying out enzymatic reactions, besides being structural, they also join with other proteins to build bigger structures that we call factories, motors, machines, things like that, names like that evoke something very complex. That's what we call the things that are made out of proteins. Here, for example, is the protein that carries oxygen in your blood. What's it called? Hemoglobin. Hemoglobin is made up of 4 molecules of hemoglobin, two with 141 and 2 with 146 amino acids. In the middle of those hemoglobins is the heme group. The heme group is a gigantic porphyrin ring that's not made out of protein. And in the middle of that ring is a molecule of, what do you take if you're deficient in iron? That's right. That's what the iron that you take is going for to make up hemoglobin. That hemoglobin picks up oxygen in your lungs. It goes to your tissues. As the oxygen level in the blood decreases, that protein changes its shape, which makes it easier for oxygen to get off. Until finally it gets to the very end, it finally releases the last oxygen where the gradient is the steepest, so that everybody gets some oxygen throughout your body. All right, back to the origin of life. In order to make a living cell, we've got to have a lot of proteins. Anybody have an idea how many you might need? At least hundreds of proteins you would have to have in order to have a living system. How are you going to get from a bunch of amino acids that have the wrong-handedness and are missing some of the amino acids? How are you going to get from that to 400 or 800 proteins that all have to have specific shapes so they have specific functions? Anybody here have an idea? You know, your lack of hands is universal. It's not just you. You can ask that question of the most intelligent scientists in the world, and you'll get the same answer. We don't have a clue. So a typical protein of 250 amino acids, that's a medium-sized protein. We have 20 amino acids to choose from. We're going to put 250 of them together to make that protein. How many different ways can we do it? No idea? Well, it's 20 to the 250 power. What does 20 to the 250 power mean? Is that a number you're familiar with? Does your bank account look like that? Okay, let's see what it looks like in a minute. But that equals 10, we're more used to 10 notation. That's 10 to the 325 different ways you can put those amino acids together. How are you ever going to find the right way to put them together? So sometimes you can substitute one amino acid for another one and it'll still work. So we'll allow some leeway there. So let's say 10 to the 250 is the number of possible proteins that you can make. That's what it looks like. One protein in 10 to the 250. I don't know how to call that number. Do you? No, I don't know. Is it gajillion, jillion, billion, trillion? I don't have any idea because I've never seen a number that big before. But there it is on the screen. That's how many possible combinations there are with only 250 amino acids. How many of those will work for the protein that you need? One. If you don't have that one, you got nothing. All right? The ocean, I'll bet you didn't know this fact. The ocean has 10 to the 21 liters of water in it. 10 to the 21. I don't know how many billion, trillion, zillion that is, but that's a big number. If we put 1 billion bacteria in every liter of ocean water, you better not drink it. Well, you shouldn't drink it anyway, but okay, and if all those bacteria, those billion bacteria in every liter of 10 to the 21 liters of ocean water, if every bacteria makes one of those proteins every 20 minutes, is it 20 minutes? Yeah, 20 minutes. So every 20 minutes, one of those bacteria tries out one of those possible 10 to the 250 proteins. In all time, that'd be 10 to the 14 generations, you could have 10 to the 50 tries at making the right protein. Does that help? It doesn't help at all. What's the chance of you're having the right protein with only 10 to the 50 tries? 10 to the 50 tries, that's a lot of tries, isn't it? But you're nowhere near where you need to be. All right, let's try one more big number and then we'll move on. If the history of the universe is 10 to the 17 seconds, and there are 10 to the 80 particles in the universe, I checked both those figures on my Ask Google phone tonight, and they're still correct. Okay, so there's 10 to the 80 particles in the known universe. That's a big number, isn't it? That means you count up all the protons, all the electrons, all the neutrons, and all the other particles that are out there. There's 10 to the 80. There's 10 to the 17 seconds in the history of the universe. If you use the reverse Planck's constant for the speed at which reactions take place, there are 10 to the 44 possible reactions that a particle can have in 1 second. So 10 to the 80 particles, 10 to the 44 particle actions per second, and you have 10 to the 17 seconds, you can only try about 10 to the 140 times in all the history of the universe with all the particles trying to make amino acids into proteins. Do you see how absurd that is? It's impossible. I'm not ashamed to say it's impossible. As scientists, we don't use that word. We say it's improbable. But there is a point at which it becomes improbable that impossible is not the right word to use. To put it another way, given all time, all particles, we could only have tried out an infinitesimal fraction of all the possible proteins to try to make that one protein we were trying to make. Now, if you were trying to make one protein that way, what happens when you come to the next protein you need? You got to start all over again. The probabilities are the same for the next protein, and so on. So I don't hesitate to say impossible is a good word to use. We don't have either a system to put the amino acids together into proteins, and we don't have the amino acids to do it with. Where are we going to get them from? But let's suppose that we do have the system, we do have the time, we do have the amino acids, and we want to put them together to make proteins. Let's make it easy. Instead of 250 amino acids, let's make a small protein with 100 amino acids. Okay? We're going to be happy with 100 amino acids in a protein. In 1999 or thereabouts, IBM decided if we can predict the shape of a protein with 100 amino acids correctly, we will own the world. Why? Because then you could design enzymes to carry out reactions and do all the things that you'd like to do in the world. Do you understand their motivation? They wanted to be God, really. But we all have that from time to time. So, in 1999, they set out to build a computer. If you understand the three-dimensional shape of the protein, you own the protein. So they named it Blue Gene. And their goal was to predict the shape of the protein from the amino acid sequence. So they built the largest computer that was ever built. It was called Blue Gene. Kind of a play on words. Big Blue is IBM. They're working with genes. Blue Gene. Okay, so here it is. This is the structure of the computer. You can see the chips they put into sets. Those sets they put into racks. And those racks, there were 64 of them, and that made up one computer. It had 640,000 processors, I believe. They took that computer and they dedicated it, after spending all of these millions of dollars of their profit, they dedicated that computer for one whole year to try to solve the question of how you make a protein or how you predict the protein shape from the amino acid sequence. Massively parallel processing, the biggest computer that had ever been in existence. 1 year. At the end of the year, nothing. They knew nothing. They hadn't solved the problem. So they built a bigger computer. This one was called Sequoia. It consisted of a bunch of chips and a bunch of drawers and a bunch of racks. And here's what it actually looks like. 1.6 million processors, massively parallel, each of them with its associated memory, running at 20 petaflops. They invented the petaflop name. But by then, guess what? The NSA was buying their computers too fast for them to spend a year trying to solve that problem. So they never got back to the problem. And it still sits unsolved today, except for this. Some scientists said, oh, let's not try to do it from first principles. Let's look at the structure of the protein, see which amino acids are involved in which shapes, and then try to predict what the shape of a series of amino acids is going to be. And they could do it. So by taking nature and using nature to predict, they were successful at being able to predict the shape of a protein from its amino acid sequence. But they cheated. They didn't do it from first principles like IBM tried to do. The world's most powerful computer working for an entire year was not capable of predicting the shape of a single small protein molecule from its primary structure. Yet, otherwise, do you see what that means? You see how ridiculous it is to think it could happen by accident when you can't even do it with the biggest computer in the world working for a whole year. Yet otherwise intelligent beings believe that specific functional proteins arose by a cosmic accident. And while we're on the subject of computers, this is an aside that's going to take us through the rest of the lecture. The fastest supercomputers were thought to be able to cover only about 1% of this thing up here. 1%, Sequoia. Recent studies have revealed that human brain, composed of about 86 billion neurons, is not like a giant computer, but each neuron is itself like a personal computer, making decisions and carrying out operations. So it's like 86 billion computers in your brain. And that's the same as 60,000 Sequoias in your head. So think about that when you're feeling heavy having trouble walking around, remember you've got 60,000 of those giant supercomputers in your head working away trying to get you out of trouble. That's not all. About 4 years ago now, it was found out that a single neuron, one of those, what was it? How many? Forgotten the number. 86 billion. One of those 86 billion neurons in your brain is capable of producing a whole bunch of microcomputers. Here's what it looks like. This is in the brain of a fly. Those are the compound eyes on the side, and there you can see the part in the orange square where this structure is found. And here you can see the structure. It is one neuron. It has 1,400 endings in the optic nerve of the fly, these omicron cells. 1,400 endings, each of which behaves as a single computer inside one neuron. And you got 86 billion of those running around in your head. I know, you're thinking, if I got 60,000 of those things in my head, how come I have so much trouble computing? Yeah. So if a single neuron can carry out the function of 1,400 individual computers, and we just now found this out, what else do you think we might yet find out about the complexity of the brain? Anybody with me that thinks there may be a whole lot more in there? This begs the question, how could such a complex system exist without a designer? We're not talking mush. Oh yeah, I know some of us. We're not talking mush. We're talking supercomputers inside your head. Miniaturized exquisitely on a scale that's unfathomable. The answer is clearly it cannot exist without a creator. Let's consider for a minute what the naive brain can do. How many of you have ever seen a giraffe born? Not too many? Think about this. You drop 7 feet to the ground into a pile, and I mean a pile. You get up, you start walking, and you go and find your mother's milk. Who told that giraffe how to do that? How does it know how to walk? Did you know how to walk when you were born? No. If you fill out, you'd still be there. Okay, so number one, how does it know how to walk without some circuitry already wired in the brain? Obviously, there is pre-wiring in the brain. On the other hand, human beings don't get up and walk around. But what do they do? You ever seen a mother smile at a little baby and the baby smiles back? You ever seen that? How does that baby know which muscles to contract to make its lips go up? And how does it even know it wants its lips to go up? It's built in to the brain, the circuitry to make that happen. I could say 1,000 other stories, but no, just one more. A bird. You can take a baby bird, you can take an egg from a mother, isolate it from the mother, hatch it in the incubator, raise the chick, and then when it gets old enough to sing, what does it sing? It sings its mother's song. How does it know what song to sing? Not all birds do that, but many of them do. How does it know which song to sing? Because it is pre-wired in the brain. There's no other explanation. That's all there is. Who's doing all this pre-wiring? Well, we know a little bit about that. In your ear, the cochlea, there are a series of cells along the bottom of the cochlea that allow calcium ions to go inside and make a impulse in that cell. Along the length of the cochlea, if you were to unwind the cochlea, it'd be about so long. Along the length, there are 980 different proteins that allow different frequencies of sound to activate the cell that they're controlling, so that you hear the whole range of sounds. Not just the high notes, not just the low notes, you hear the whole range of sound. 980 different proteins, all made from one gene. We'll talk about this a little bit later. One gene can make 980 different proteins that will be expressed along the axis of that cochlea that will enable you to hear the whole range of frequencies. There's a protein called DSCAM. It would be a distraction to tell you what that stands for, so I won't. DSCAM protein can make 19,800 different isoforms from one gene. One gene, the DSCAM gene, can generate 19,800 different proteins by selective joining together as parts of the gene. And it is involved in the programming of the brain. So apparently what happens is one of those isoforms is expressed in this cell. That cell then gives rise to the next cell. The next cell looks back and says, oh, that one had this. I'm supposed to produce this one according to the directions I'm reading here. And then that cell produces that one. Then that cell produces another cell. And that cell looks at the direction and says, okay, I'm supposed to produce this one. That's the only way we can explain the wiring of the brain. There has to be a superset of instructions that tells those cells how to express the protein. And as it walks along, leaving cells behind, it's programming and planning these circuits that allow us to be able to do the things we do by instinct. Let's take another look at development. How do complex systems develop? Okay, I'm guaranteeing you this. I'm guaranteeing this to you. You can check it out for yourself. Every single one of you started out as one cell. You didn't start out as a head and a backbone and legs and arms. You started out as one cell. Not only did you start as one cell, everything else around here started out as one cell. Please explain to me how one cell became me. What kinds of operating systems are necessary to govern the developmental process so that one cell in cell division can become me or you? Explain that to me. In fact, better yet, go to your drawing board and come up with a book telling me how to do this. You think you could do that? This is an egg of a salamander, or a newt. And we're going to watch it as it divides. I want you to notice, first it makes 2 cells divided evenly. Then it'll make 4 cells divided evenly. The third division is apolar. It's one of the poles away from the other pole. So we now have big cells at this side and smaller cells up here. This side's going to become a tail. This side's going to become a head. And from then on, everything is starting to be determined. Okay, watch carefully. I'll make a few comments as we go. There's the first division. Second division. Third division. See, it's eccentric. Now this is going to be the head. The other side's the tail. That's all predetermined in the egg. Just watch as cells get smaller and smaller. The eggs staying the same size, so the cells are getting smaller. There you can see them dividing. This is time lapse, by the way, so this isn't real time. Here comes the blastula stage, or the gastrula stage, sorry. And we're having gastrulation. That imagination is going to make the intestines. This is going to be the mouth, and at the other end will be the anus. So that's already being formed here. Now we're forming the neural shield. Well, not yet. Okay, there's the neural shield. That's where the nervous system is going to form. You're going to see the nerve cord right there. And at the top, we have what are called the neural crest cells. Neural crest cells are going to be amazing in what they do. Neural crest cells come from that white band that encloses the spinal cord. So that's the spinal cord being formed there. Now we've got the neural crest cells. We're going to zoom in so you can see those. Look, those cells are migrating. You see them? They're moving to places they have been predestined to go. How do those individual cells know where they're supposed to go? They're the cells that will make up the parts in the adult's body, like the spots, the melanin containing spots. Those are all formed by those cells from the neural crest migrating through the embryo, developing embryo, and going to certain specific spots. How do they know where they are? Who's telling them where to go? Who's in charge? But I want you to notice we're making out of a single cell an organism with all the organelles and all the complexity of that organism. Let's go ahead now and we'll see it continue to develop. Here we're beginning to see the limb buds, the head, the eye is popping out there on the side. There's the tail. You ladies that have been pregnant, you now understand why the baby's jumping around in there doing these stretches. Look at the little black cells. Those little black cells all migrated to where they are from the neural crest. A lot of other things as well. Here you can see the heart beating, the blood flow. And you can see they have gills, external gills, and the gills are picking up oxygen. There's the heart. There's a problem. He's caught in a very tough protein membrane. So how's he going to get out? Well, you watch, you'll see his head is releasing some enzymes. Remember, enzymes are made of what? Proteins are made of amino acids. Specific enzymes that digest that particular membrane. See how thin it is now? You can almost not see it there in front of his face. There he goes. Did he break that membrane? No. It dissolved at just the right time for him to get out and be on his way. That happened by accident. That happened by random processes over billions of years. Or is there an immensely intelligent designer behind this? I mean, everybody's free to make your own decisions. I'm not trying to force anybody to believe anything about this, except be reasonable. All right? Now let's look at molecular biology. All the complexity of modern organisms is found in the very first organisms we find in the fossil record. If we go way down to the Cambrian, to the very first metazoans, we find out that some of them are still alive today. We can look at how complex they are, and we can answer the question, were they fully as complex as anything today? And the answer is yes, fully as complex. No proposed mechanism for the origin of life and complexity can stand careful scrutiny. I say that without apology. Nobody can come to me, nobody has the wherewithal to come to me and tell me that they understand how life originated from non-living material, because we don't have a clue. We've made some efforts to try to understand it in naturalistic terms. Sure, we can make a few amino acids. We can even make those amino acids join together to make proteins under the right circumstances, which are different than the circumstances that made the amino acids. We can do those kinds of things, and we can play that game. But you still cannot tell me how you make even one single protein without a living system. Nobody can tell me that. All right, let's look at the Human Genome Project. The year is 1999, the year 2000. We finished sequencing the human genome. For the first time, we knew how many genes were present in human beings. And we were shocked because there were only 20,000 genes. Only 20,000 genes in humans. Well, there are more than that in the turnip. And it didn't look very good for us. But we know there are 100,000, at least 100,000 proteins expressed in human beings. With 20,000 genes, 100,000 proteins, how do we get from one to the other? I've already given you a clue when I told you about the DSCAM protein, that you can take these genes, cut them up, put them together in a different order, and make a different protein. And if somebody has designed it to do that, then that protein will have a function, and that function can be expressed in the living organism. So we soon discovered many genes make more than one protein, combining in different ways. But how do you do that? You have to have a sophisticated control mechanism that overrides the normal level of complexity of the genome. You have to have another layer of complexity up here that determines which way those things are going to be put together. And then you'll need another level above that to tell which one of these to be active. You see what we're getting here? It's not a matter of Darwinian process producing a new gene that does something good for an organism. It's a matter of some super intelligent persona, who we call God, using brain power that we know nothing of to cause these things to happen in the right way. So let's take a look at genetics. Here's the way we looked at a gene when the Human Genome Project was completed. This is what I taught my students. I taught them that one gene makes one enzyme. You know, a lot of things that your teachers taught you are not true. Probably some of them more than others. But this is not true anymore. We used to say a region of DNA giving rise to one protein was a gene. Then we discovered splicing, and here's a little video that shows how splicing works. Here you see a DNA strand in pink, and the RNA strand is being produced by that DNA polymerase, DNA-dependent RNA polymerase. It's making the RNA, and then a series of proteins, about 30 different proteins, comes together on that RNA and decides which parts to keep and which parts to cut out. And here you can see it's cutting out the green part and joining together the two yellow ends to make something that will go out into the cytoplasm and make a protein. So DNA is the master control. RNA is the functional driver of protein synthesis. And so here you see making an RNA by cutting out pieces and throwing them away or doing something with them. This one's going to release it, and then it's going to be dissolved. Okay, here's a RNA that's been copied from the DNA. So the DNA is the master copy. The RNA is the functional copy. We're going to throw that RNA away after we've used it. So we're just going to splice out the parts we don't want and make a functional RNA. Now what we found out is that some of those parts are put together and some are left out. So you synthesize the RNA, then you chop it up in pieces, put those pieces together in different ways to make different proteins. Now that's not haphazard or random or accidental. Each of those proteins is predestined to be. And there is a control in there that says, this time I want you to make this with this. Next time you make this with this. And join those together to make a different RNA that will go out in the cytoplasm and then be copied in the ribosome to make a protein. So that's the information on how to make a protein right there. Here's what we look at a gene as now. Remember the other one was just a stretch like this, just a line. Now we know all these other functional activities are carried on by that gene. And we won't go through the details of them, but the one part that I will point out is the pink parts down there at the bottom are the parts that are going to go out into the cytoplasm and be copied as a new protein. So the other parts of that are all involved in regulation. How do I turn this on? When do I turn this on? How many copies do I make? What do I do with the copies? What kinds of alterations do I make to make it what I need? All that is encoded in that, what we thought was just a stretch of DNA before. Wow, we were so naive. So, I told you already, there were only 20,000 genes in the human genome. What is all the other DNA doing? In fact, 20,000 genes take up only 2% of all the DNA. 98% of the DNA is not genes. So evolutionists glommed onto that and they said, okay, this is DNA that's evolving. So here's where the evolution is taking place on this DNA that's not genes. That's what's going on here. We understand it. We called it junk DNA. Junk DNA is the DNA that's not genes, that supposedly is just evolving into something else. I didn't like that idea, and I said so at the time. In 2000, shortly after 2000, the U.S. government funded a project called ENCODE. The purpose of that project, and all those millions and billions of dollars of your tax money, was to show whether that 98% of the DNA was doing something or not. Because we have quotes from Richard Dawkins, the evolutionist, and other people saying, I could do just as well without 98% of my DNA. And I say to him, great, go for it. Because we're going to find out in a minute, that's not junk DNA. So they developed tests to determine whether that DNA was being copied, being read, or being used. And after a few years, I think it was about 14 years, I don't think I have the date in there. After about 14 years, they came out with a series of papers, 20 papers all at once by 400 scientists in labs all around the world that were studying what that junk DNA was doing. And the conclusion was at least 80% of that DNA was functional. And they didn't really know what it was doing yet, but they knew either that when they cut it out, the organism died, or it didn't thrive, or it made a difference in what the response was. It was doing something, 80% of it. They didn't know about the other 20%. This is an analogy. This is a machine shop, a very orderly machine shop, one that I wish I had. A machine shop, an orderly machine shop, has drawers. And in those drawers are the things that they need to make stuff. So a machine shop has all the stuff in it, in the drawers, but it's not doing anything, is it? It's just sitting there. In order for that to be functional, what has to happen? We have to have a plan, right? How much information is in a plan? A little bit or a lot? A lot. It involves the design. It involves the information about how to make that design, to actuate that design. That's all in the blueprint. What else does it require? If you have a machine shop and you have a set of plans there and you have all the drawers, what else do you need? A machinist. That's right. You have to have a machinist, because without the machinist, the plan will never be actuated. Without the drawers, the plan will never be actuated. They all are needed. But what were we looking at as the whole? We said the drawers are everything. That's the genes. The drawers are nothing without the plan and the machinist. What's the plan and the machinist? Well, we better look deeper into that 80% of the DNA that's not genes, hadn't we? The Uncode Project has liberated us forever from the possibility that we are the result of some cosmic accident. Clearly, we have a master planner who has encoded our DNA, in our DNA, the operating system, the blueprints, and the directions for proceeding, and the machinist to accomplish the goals. It's all there in the DNA that we thought was junk. What do you think of that? What were we thinking? That genes by themselves were going to run the machine? How could we be so stupid? I include myself. This is molecular biology as a whole. How could we think that genes were all you needed? You got to have a lot more than genes, and the things you need are information about how to make the genes function in the right way, and then you need a mechanism to carry that out. So all this has to be involved in the genes. And then we thought we were through, but nature wasn't through with us. Dogma was that genes were everything. And the environment had little to do with development. I remember when I was teaching my students in undergraduate biology, and I would talk to the students about the effects of their behavior and their lifestyle on their offspring. Because we knew that women carried with them all the eggs they would ever produce in their life. And so if they did things that were bad for their body, what was going to happen to the eggs? They're going to be affected. And I said, out of an understanding of fairness, I said that can't be the whole story. There has to be an effect on the males. Their behavior also has to come into this equation. You can't blame your kid's behavior just on the mother. It has to be fair. And for many years, I didn't know what the answer was, but I told my kids, don't think you're going to get away with this. If you behave this way, you're going to affect your offspring. Then this paper came out in Nature, the most prestigious biology journal in the world. And what's the title? Epigenetics, The sins of the father. Where's that phrase come from? Somebody. The second commandment, did I hear that? The second commandment, the sins of the father will be visited on the children to the third and fourth generations of them that hate me, and showing mercy unto thousands of them that love me. And remember that second part. That's really important, because all of us have messed up. But God hasn't thrown us away. This is a picture from The Simpsons, the sins of the fathers. The experiments were these. They took mice and they exposed them to a pleasant odor. They used cherry blossom smell, which is very pleasant. And when they exposed them to cherry blossom smell, they would shock them. Shocking, isn't it? And that's the way they trained them. So that then they could expose them to the cherry blossom smell and the mice would run. So they conditioned them to run when they smelled cherry blossom smell. Then they bred the next generation of mice together. And they said, I wonder if somehow these mice are going to respond the same way. And lo and behold, they did. So they took that generation, they bred them together, and they said, I wonder if they're going to express it in the third generation. And they did. And finally, between the third and fourth generations, they would lose that adaptation. Let me ask you this. You are a competent designer. You're an engineer. You want to transfer some information from a parent to an offspring so that they will remember. Give me a way of doing it. Show me how you would express cherry blossom smell that the mouse had never been exposed to before in their life. How would you teach that mouse to remember that smell and to react to it by running? How sophisticated a mechanism is this for transmitting information from the parents to the offspring? Wow. Scary, too. I want you to watch how DNA is tied up. This is a DNA strand. A DNA strand is very rarely isolated by itself. It's almost always tied up with special proteins called histones. And you're going to see some histones glom onto that DNA and wrap the DNA around themselves. And pay special attention to those tails sticking out. Do you see the little blue wiggly tails? Those are the part we're going to talk about. Are they DNA? No, they're histone proteins. They're tails on the protein molecules that the DNA is wrapped around. And here you can see how those histones cause that DNA to fold and fold and fold and fold until it becomes a chromosome. Pretty amazing, isn't it? And so far as we know, they've never seen a little man in there telling you what to do. So this is all self-governed properties. And here we're going to watch the cell divide just for the fun of it. There are the chromosomes all lined up at the equator. Now on a signal, off they go to opposite sides to make two new cells. Pretty amazing. Okay, back to our question. How do you program into the offspring information to recognize the smell of cherry blossom and to run? Any ideas? Okay. Do you see the tails on those histone proteins? Those tails have amino acids in them that are bound by either a phosphate molecule or a methyl group or an ethyl group. And so they are they are tagged with information that determines which genes are turned on and which genes are turned off. This is called epigenetics. It's controlling the genes not by the protein, not by the DNA, but by the protein that binds the DNA. How clever is that? Who would have thought of something like that? And how do you decode this information? Because you can see it's just like keys on a piano. You're going to play them this way or you're going to play them that way. Each one makes a different tune. It's all built into the system. The cell then contains an additional sophisticated higher level of regulation that was unimagined until recently. We really never got our eyes on this until about 2016 when these studies came out. The mechanism itself has to be controlled. Not only do you have the control mechanism that plays the piano, but something has to say how to encode that. So there's a higher level still above that that determines this regulation. I don't know. Have you ever heard an evolutionary explanation for this? How can there be? It's so sophisticated we can hardly grasp it. We certainly don't understand it. And we certainly, even though we know how these encoding work, we don't know how you encode cherry blossom and run and then attach that to the proteins and then let that control the DNA. Which DNAs? To make the mouse able to respond. And we thought we were done with complexity, and then we discovered even more. Orphans. What are orphans? Orphans are genes that are found in humans that are not found in chimpanzees, which is supposed to be our ancestor. Orphans are genes that are found in honeybees, but not in other bees. That's what orphan genes are. They don't have any parents. Doesn't seem to be any place they could have come from other than by creation. How did the bees get the genes to make honey, and they don't go to the wasps or the solitary bees or anybody else? It's only the honeybees. How do they have genes in them that nobody else has? We already talked about the impossibility of generating even a single protein by random processes. It can't happen. We can't make that gene for making honey just by accident, serendipity, or just putting things together. Where did the genes in the honeybee come from? There's only one answer. It came from the originator. The originator of innovation. In humans, about 500 genes that we know about are orphan genes. They don't have anything in the chimpanzee that could have given rise to them. If we think we came from the chimpanzee, we have to come up with an explanation for those, at least for those 500 proteins, those 500 genes that do important functions like make our brain bigger. One of those genes is responsible for our bigger brain. That's the state we find ourselves in today. It's not a happy state for people that believe that this all happened by chance. It's not a happy state for naturalism because it begs the question over and over again, where is the great cause of all this? And do you think these people don't rack their brains trying to explain some of these things that are unexplainable? Evolution by random processes and natural selection lacks explanatory power for the existence of even one protein. That's a harsh statement. No conceivable naturalistic explanation can help us to understand the human brain. You know, go to your drawing board tonight, come up with a solution by tomorrow that tells me how you make a human brain without outside help. The genetic discoveries of the past 20 years defy explanation within the evolutionary paradigm. Evolution was a fine theory back in the Darwin's day when we knew nothing about the cell. The cell was just a bag of fluid. Could it happen by accident? Maybe, I don't know. But this is not Darwin's day. We have very sophisticated understanding of some of the processes of life, and it's not looking good for evolution. Thank you.
Wow. Thank you, Dr. Chadwick, for taking us on a journey of discovery within the cells and the molecular biology that makes us today. You were made in the image of God. You reflect your Creator. God has purpose for your life. If you think this was an interesting start, you don't want to miss tomorrow. I understand we're going to be hearing about the fossil forest in Yellowstone. Dinosaurs, who likes dinosaurs? Tell you what, I'm looking forward to tomorrow. Do not miss tomorrow. It's going to be just as good. And this scientific discovery is a journey. It's a journey of, it's an adventure that we get an opportunity to go on with Dr. Chadwick. Don't miss tomorrow. Make sure you're here. There will be lunch tomorrow served between the first two presentations and the last presentations. So there will be good food. If you have not registered, please see our registration booth on the way out so we can know how many to prepare for. Lunch is free. We are delighted that you all are here, and we're honored. Let's go ahead and say a prayer and close off. Precious Lord, we want to thank you so much that we are fearfully and wonderfully made in the very image of God. Lord, our own attempts at trying to explain how life came to this earth without you are mere rubbish, Lord. We recognize that you are the great designer. And Lord, we're excited to learn more about what science has to say in support of that. Because, Lord, that's what gives our lives purpose. That's what gives our lives hope. And that's, Lord, what gives us a scope and a vision for eternity with you. Please bless each family and each home represented here tonight. And thank you so much for this opportunity we have to learn from Dr. Chadwick. In your name we pray. Amen. These programs are going on onto YouTube and also are being live streamed.